During the first phase of meiosis I, prophase I, chromosomes begin to condense, and the maternal and paternal chromosomes that form each homologous pair line up next to one another in a process called synapsis. Each complex of 4 chromatids produced by a homologous pair of chromosomes lining up next to one another is called a tetrad. Networks of protein strands join the homologs in each tetrad together, shifting their positions relative to one another until the corresponding genes on the homologs line up exactly with one another. After the genes of the homologs are lined up, special enzymes exchange corresponding sections of genes between the 2 homologs. In the course of exchanging these sections, the paternal and maternal chromosomes come to cross over one another at points along their length, called chiasmata. Besides reflecting where chromosomes have exchanged sections of genes, chiasmata also hold homologous pairs together until they are later separated. The exchanging of sections of genes between homologs is appropriately enough called crossing over. Crossing over, by creating chromosomes with unique combinations of genes, is one of the events that occur during meiosis that help increase the diversity of individuals within a species. While homologs are lining up and crossing over in the nucleus, outside in the cell cytoplasm, other processes critical to meiosis are also taking place. All eukaryotic cells have a structure called a microtubule organizing center, or centrosome, near the nucleus that synthesizes the protein tubulin. Tubulin is used to create long, thin filaments called microtubules. A network of these microtubules radiates out from the centrosome, helping to form and maintain the cell's shape. During meiosis, the network of microtubules radiating out from the centrosome disappears, and the centrosome divides into 2 new daughter centrosomes. The daughter centrosomes then move to opposite poles of the cell, each forming a network of microtubules that interconnect with the microtubules radiating from the other to form a structure called the spindle. The spindle consists of polar microtubules that radiate from either centrosome and overlap at the equator, and kinetochore microtubules, which will eventually attach to the chromosomes at their kinetochores, which are located near the centromere where the 2 sister chromatids are tied together. Once the centrosomes are at opposite ends of the cell and the spindle is completely formed, the nuclear envelope disintegrates, allowing the kinetochore microtubules to break in and attach to the chromosomes. Due to the lining up of chromosomes crossing over, and formation of the spindle, prophase I is by far the longest stage of meiosis, often accounting for over 90% of the time spent by cells in meiosis.